Thanks again for the invitation. I'm really happy to talk about my work, and I think your introduction was basically covering everything I would say myself. Right, so just briefly, I'm in the computer graphics and visualization group, and really here my background is working on visualization, visual analytic tools, with a focus indeed on high-dimensional data. We've done quite a bit on spatial data, so spatially resolved high-dimensional data. Throughout this talk, I will not only show applications from biology. One of the reasons is that my demo computer crashed this morning, and I have to resolve or revert to video demonstrations. Unfortunately, I wanted to give live demos, but I will show a bit like these methods on different applications just to give you an idea what is possible in other areas. And actually, most of these things can be transferred also to say spatial biology data. So I guess I don't have to introduce much, but high-dimensional data, right? So up until not so long ago, when we thought about high-dimensional data, we were mostly thinking of like tabular data, abstract data, where we have, for example, what I have here on the slides is a single cell table, where every row is a cell, and then we have protein measurements. Every column is one of these proteins. Could be genes also, these kind of data. So we get these huge tables with many rows and many columns. But in recent years, right, we got all this spatially resolved high-dimensional data. Like we have this multiplex tissue imaging that you guys over there are very well aware of. But these data are also available in other domains, right? So satellite imaging has been using like hyperspectral measurements for quite some time. So the idea here is instead of only getting RGB for an image, right, you get actually a full spectrum of the visible or it's also invisible light, right? So you basically also get a high-dimensional space there per pixel. I also have collaborations with in art and history, so with the Rijksmuseum here in Amsterdam, and they acquire imaging of paintings in a similar way. So what you see on the bottom right there is X-ray fluorescence data. So basically, what they do is they shoot an X-ray at these paintings, and then this X-ray activates the electrons on the outer shell of the different elements, and depending on the element, you get a different response, fluorescent response, and that then we can measure so that we can see, like in this example, that really images for different elements like iron or copper and so on. Right, so we really have this high-dimensional data in quite a few application areas. And so we are actually trying to look at this also from the method side on how can we actually improve the exploration of such data. Now, when we think of how do we visualize high-dimensional data, well, like the traditional methods are kind of what you see here, like parallel coordinate plots or scatter plot matrices, but they are kind of limited in the number of dimensions we can look at. So what has been sort of the go-to tool in the recent years is basically using dimensionality reduction for visualization, right? So you have some high-dimensional data like you see here on the left. I tried to indicate the 4 dimensions, but actually, right, so every column of your table would actually be a dimension here. And then the idea is that we try to reduce this to just 2 so that we can actually see it on a computer screen, right? So by doing so, obviously, we lose some information depending on the approach we choose, right? A different kind of information, but we can get some 2D representation that keeps some information, some features of the original data. Following Bremer et al.'s paper from 2014, basically there's 2 major tasks for dimensionality reduction, and one is dimension related. I have grayed these out here in the slide. And the other ones are more cluster related. So looking at verifying clusters, naming, matching clusters, these kind of tasks. And they are very much related also, for example, in biology, that we would want to identify groups of cells, right? So there would be a cluster in these plots. So we want to identify what kind of cells those are and so on. So when we want to really identify clusters in high-dimensional data, well, then we have to make sure that we choose a dimensionality reduction that actually preserves clusters. And I'm sure you're all aware of TSNE and UMAP, right? So these are basically the two tools of cluster preserving dimensionality reduction or neighborhood preserving dimensionality reduction, right? So the idea of these is that you compute the neighborhoods in the high dimensional space, and then we model a lower dimensional space that tries to match these neighborhoods in the 2D space. Now, the issue with these is, well, even UMAP, they are computationally quite intensive, especially when we look at like imaging data where we have hundreds of thousands of millions of pixels or even more, right? So I know you have these crazy data sets that are, I don't even know how big they are, but right, so they don't even fit on a disk. So in this first part of the talk, I would actually look at what we did in quite a few years ago, actually already on improving the performance of these tools such that we can actually use them in sort of explorative visualization settings, right? So that we don't have to wait a day to get such a plot on the right, but actually can use this while we work with the data. So I want to show, since Niels also already mentioned Cytosplorer, I want to show a very brief video of what this kind of cell type identification or cluster labeling in Cytosplorer would look like. So here you have basically the main interface of the tool. We have this TSNE plot in the middle, right? So each of these groups of points here is a group of cells that were positioned there according to the similarity of the cells in the high dimensional space. So this is mass cytometry data. So we have actually protein measurements, right? So you see all the proteins here on the left side. And then, right, according to all of these selected dimensions, then the plot is calculated. Of course, right, so very common, you see then now the coloring here by a protein, right? So that's sort of the standard approach. We show one of the proteins now as color. On top of this, I can cycle through a few of these different dimensions just to get a different impression of the data. But most importantly, right, we want to look at actually what's happening here in these clusters. So what is, or in these groups of cells, what is that type? So we have sort of an interactive way to extract the visual clusters from this map. So I should stress this, that this is not so much really for clustering the data, right? So you might want to use an offline tool for that, but rather for exploration to just explore what we actually see and then get a first impression of what's actually in the data. So in this little sequence, I just selected a few of these created clusters and I merged them so I get sort of a higher level picture of what's going on in this group. And then I can just show them on the fly here in the heat map, right? So, and you see as I move over the heat map, these clusters get highlighted on the plot on the left. I can select these and really get a bit of an idea of what's going on, right? In addition to this, we can also look at the samples, right? So that's something you probably want to do early on to make sure that you don't have something like this, where really the samples are sort of splitting the plot. Rather than the actual cell types, right? So you would really want to have a plot where these are much more intermixed, showing you that your samples are actually working well together. So that was just really a very quick high-level dive over this tool, right? But I hope you get the idea that we really want to have sort of these interactions directly between these different views, right? We want to see what's in a cluster, what do we actually see there so that we can actually interpret the data. But as I said before, right, if we now want this in an explorative visualization setting, then especially the TSNE becomes a bottleneck if we want to compute it quickly, right? So in this first part, I really want to look at now how do we make this fast? And what you see here already in this animation, right? So this is a TSNE calculated, actually not live calculated, but live captured the calculation. So it takes like 10 seconds to calculate these 80,000 data points, which, like, if you look back a couple of years ago, that was quite the feat to get there. So what is actually expensive about TSNE? Right? So you basically have two major computation steps. So the first step is computing these high-dimensional neighborhoods. And I don't want to go too much in the math here, but in principle, for every point pji, you calculate the probability that the point pj is actually in the vicinity of a point I, right? So for every point in your data set, you have to look at all of the other points, whether they are in the vicinity of this point, right? For that, you calculate the distance between these 2 points. Well, that's what you have here on the top. But then in TSNE, we normalize this by the distances here over all of the other points, right? That's actually one of the advantages of UMAP. They basically threw this normalization out, which means they actually can reduce the calculation time quite a bit. But since we have to calculate this over all of the points, now that means for every point, we have to normalize this. We have to calculate the distance to all of the other points, which means we end up here with n squared complexity if we just naively do this for all of the data points. The second part is then optimizing the low-dimensional neighborhoods. And what we do here is calculating or minimizing what's called the Kullback-Leibler divergence. So basically the distance between two probability distributions. Probability distribution P is basically all of these Pij's in a matrix. And then we have the same Q in the two-dimensional space. So we have another probability distribution here, Q, that models the similarities in the 2D space. And as I said, we want to make this as similar as possible to the P distribution so that we can actually make sure that the structure that we see in 2D is similar to the one to the structure in the high-dimensional space. And again, right, so that means we have to sum over all pairs of points, right? And again, we get n squared complexity. So we looked at both of these parts. So a PhD student in our group, Nicola Pezzotti, he actually came up with the idea quite some time ago, right? So 2017 already, to approximate the neighborhoods in the high-dimensional space instead of calculating them precisely. And in 2017, surprisingly, nobody had done that yet for TSNE. If you look at implementations today, basically every implementation does this. UMAP out of the gate used approximated nearest neighbors, and also all the other methods that you basically now get in whatever framework you use, do these approximated nearest neighbor queries. But at the time, we got from standard Barnes-Hott TSNE taking 3,000 seconds to basically doing this in 34 seconds. So 100 times speed up here on the MNIST data set, again, 80,000 data points. So not huge, but still, we really could reduce the time that it takes to get there, right? So that was the first part, actually looking at the high-dimensional neighborhoods. But now the second part is optimizing these low-dimensional spaces. And already early on in the original paper, that was presented also as not, this optimization, this minimization of the Kullback-Leibler divergence, you can think of as a force-directed layout where all points just want to be far away from each other, but those points that are similar to each other, they also attract each other. And that basically then, if you just let this force-directed layout move itself out, it will actually create sort of the space that you are interested in. Now, already the original inventor of TSNE, Laurens van der Maete, he came up with the Barnes-Hott SNE, basically with the idea that, well, large distances in TSNE, they don't matter anyways. So we can just group all the points that are far away from my current point, right? And then calculate the distances to the group instead of each individual point. So that already reduced the complexity to n log n, but still could be improved. So again, work with Nicola. He had the idea, why are we calculating actually the forces between all points? So if we calculate instead like a force field, like in the illustration you see on the right, where we calculate the forces like on this field, then and just place the points in there and move them according to the field, then we can, if we choose a resolution for that field that is small enough, get rid of a lot of computations, right? So if we assume that the number of vertices in this field is much smaller than the number of points in our data set, which is exactly what happens when we have these very large imaging data sets, then we can actually much more efficiently calculate this field. And on top of this, we don't have the approximation of the Barnes-Hut, where we actually group these points that are far away. Another nice thing about this, already in the original implementation, is that we could use parts of the graphics pipeline to actually calculate these forces, the force fields. So we could move this very efficiently onto the GPU and get also additional speed boost from really the GPU being this big parallel processor that we nowadays all use for our deep learning approaches also. So with this, we basically got this original TSNE on the MNIST dataset that I've showed you before, right? From 2 days to this 10 seconds you see on the right here, right? So really, this optimization, like with a tremendous speed up, like even with the Barnes-Hut, this was still in the range of an hour to minutes, depending on the parameterization. And here it's really something that we can just run. We don't even have to get a coffee while we wait for it. And since we also got rid of the Barnes-Hut approximation, we are actually also quite a bit better in terms of precision than Barnes-Hut and also derivative methods of this. So you see here a green line in these charts. That was a CUDA implementation, also on the GPU, that came out around the same time as our publication. And they actually just moved the Barnes-Hut to the GPU. So you see, they actually are quite a bit faster. So this is our line, this is their line. So they're quite a bit, they are a bit faster than ours. But if you look at precision, so this curve, you want this to be as far to the top right as possible. It's a precision recall, right? How many of the neighbors in the high dimensional space do we find back in the original low dimensional space? Then you see that we actually, these are all the Barnes-Hut approximations, including the CUDA version, and ours is quite a bit higher than this. And this is true also for different data sets, right? So here we have the Vert2Vec with 3 million data points, right? So we get this curve up there. All right, so that was really nice. So now we have a TSNE that actually is much, much faster than before. So let's go and move it into our interactive systems, right? So the second part that I want to focus on in this presentation is now improving the structure of the embeddings we get. And there's actually two kind of axes I'm going to look in. And the first one now is really on looking at high dimensional or highly multiplexed image data, right? Yeah. So when we look again at the formulation of TSNE, right? So that's the calculation of the neighborhoods in the high dimensional space. Then there's actually nothing that's specific about image in there, right? So we calculate the distance between these 2 points, right? That's sort of the main place where the data comes in. And this is just some distance measure. Now in standard TSNE, basically we take these 2 points, right? So these are here we have on the y-axis in this illustration, we have the channels, right? So these are 2 pixels in this image. And the distance typically then would just be the Euclidean distance, maybe the cosine distance, right? Depending on what's in the implementation that you're using for the dimensionality reduction. Now with my PhD student, Alexander Fied, we basically thought, well, can we not sort of introduce image information into this? So let's assume we have an image like this, right? Where we have black and white pixels in there. And we now do a dimensionality reduction on the pixels, right? So here on the right, you see every of these points is 1 pixel in this image. Then we get 2 groups, right? So if we do TSNE, right? So we get the black pixels and we get the white pixels. But if we look at this from an image perspective, then we actually have three groups, I would argue, right? So we have the white area, we have the black area, and then we have this checkerboard area. So we would like to have something that gives us something like this, right? Where we have exactly these two groups also represented in our T&E embedding. So what we did with Alexander was basically look at distance metrics that would give us this. So we integrated area-based distances into the T&E process. And we basically have two larger types of methods. So metrics, one is what we call it's basically a point cloud distance. So we take all of the pixels, like to calculate the distance between this pixel and this pixel, we actually take pixels around it. And then we just put all of these pixels that are in one area into a bag, and then we compare the two bags of pixels, right? So these two point clouds. Now, the other idea is we can also extract features from these, like image features, any kind of image features, and then do similarity calculation based on these. So here we would just calculate a histogram for each channel and then calculate all of the histograms between two channels and then sum up the distances between all of the channels between two pixels. And the other idea there is actually calculating features that are for the whole high-dimensional space. So here in this, we would calculate a covariance matrix and a mean vector for over all of the channels, and then just compare these two directly with each other, right? So we have these feature-based methods, and one is per channel, and the other is looking at all of the features together. Right, so here's a very simple example of how this would look like. So this is our actual data. So it's just two-dimensional data, not very high-dimensional, right? But we, just for illustration, we see that we basically have these four groups of pixels, right? We put a bit of noise on this, so it's a nice Gaussian distribution so that we don't run into like bad numerical issues. But essentially now here, every pixel is colored with the color that we assigned here. And now we do a T&E on this, and we would get something like this T&E, right? So we get the four clusters that we would expect of the four classes with using, say, standard Euclidean or cosine distance. And now for the illustration, I'm using a little trick here that I take the XY coordinate of my T&E embedding, map it to a color, and then project this color as RGB back into my image space. So basically, the color you see in this image is the position of the corresponding pixel in the T&E map, right? And that allows us to see the structure a bit. I'm going to use this a few more times in the presentation, so just that you know how this roughly works. Now, if we look at the three distance metrics that we defined, right? So this first one here, that's the per channel histogram. Actually, there the structure is not so great. I mean, we do see the separation of these 8 regions that we actually have in the image, but in the embedding, it is not. Yeah, the structure is a bit tricky to really identify. But if we look at these two here on the right, those are those that actually look at all of the dimensions at once instead of per channel. And they actually do exactly what we expect, right? So we have basically these 4 major clusters still that correspond probably to the inner parts. I'm not 100% sure right now. But then we also have smaller clusters that are sort of the other regions in the image. And then the remapping really shows basically we have our outer parts here with the checkerboards. We have the inner parts with the homogeneous values. And on top of this, we also get a bit of the boundaries in between. So yeah, it's not quite clear if we really need those, but just to be direct here, like what we see actually in this crosses, right? But generally very nice, right? So we now see actually this kind of extra image structure in our embeddings and can reason on this. So I can show a brief example here on geospatial hyperspectral image analysis. So what you see here on the top left, that is the ground truth of a satellite image data of different crops. So each of these colors is a field with a different crop. And again, the measurement is this hyperspectral data. So we have this many channels of the, I think in this case, mainly visible light. So again, if we do a TSNE, what you see here is the TSNE embedding, colored actually now with the ground truth, right? All the gray parts, the light gray parts, they don't have a label, but you see all the labels kind of distributed in the TSNE. And while we do see some structure, we also see that they are kind of spread out. So now just to make sure that with the method I just explained to you, not just smoothing the image, we actually also just smooth the image and compare this. So this is a Gaussian smooth version of the image, and then run through a regular TSNE again. So again, we see kind of the structure, some of the clusters, like this green one over here, right? So they are quite nice, but then others are again a bit more distributed, like if you look at these blue ones over here. And this is basically, again, the version with the point cloud distance. And now you see really compact clusters, right, with in brown here, in blue. There's the orange ones on top of here that really map to the structure of the actual data. We also looked at this a bit more quantitatively. So basically calculating here the neighborhood hit over k neighbors. So basically we look in the 2D space at k neighbors and then look at how many of these neighbors actually do have the same label from the ground truth, right? And you see that the regular TSNE gives you like constantly below 50%. Then we have the smooth version here, and ours here actually, while it's dropping with more neighbors, gives actually quite a nice precision, especially right in these smaller neighborhoods. And again, this remapping of the color now shows nicely also here that we really extract more high-level structures like these fields compared to the very noisy images you see on the left, right? Where you really see that points from different areas on the embedding like are close together now in the image space. Okay, as I said, I don't have these live demos. I would have loved to show you something about paintings here, but I'm just showing you these now again a bit in an interactive fashion. So this is not in Cytosflow, but actually a new framework that we based on this. It's called ManyVault. It's more of a general exploration framework for high-dimensional data. And what you see here is actually, again, these three embeddings. You see the data, these fields here on the top left. So, and I'm just going to do a few selections here to show actually where these clusters show up, right? So again, this on top here is our spatially where embedding, and you see like I'm going through selecting in different places. I'm now showing the ground truth on top of this. So if I select now here in the image, then you see how nicely, again, this maps to a cluster over here and how it's distributed in some of the others, right? Okay. So that was the one part of improving the structure of the TSNE, right? So really focusing on image data, trying to get the image information into the TSNE. And now I want to look at a different angle and actually looking at large data. So what does happen actually when we put the really large data in TSNE? So this is a TSNE plot of 5 million cells, imaging mass cytometry, no, not imaging, just regular mass cytometry. I calculated this like 6-7 years ago, before we even had the GPU version. It took me actually a week to compute this. And you see actually there's very little structure that you see in this, right? So we see the sort of these big high-level blobs, but otherwise there's not much going on that we can really reason on. So there's two reasons to this, I would argue. One is that TSNE is just not very good at pushing sort of the points at the right direction, places when there's a lot of other points in between. And the more points we throw into this data, the harder this becomes. And the other one is just visually that when we throw 5 million points in a scatter plot, well, and we have only, say, 2 million pixels on the screen, then at some point we also run out of actually ways to show all these points, right? So I also calculated versions of this, just taking random samples of the data set and then calculating a t-SNE on these. And you see with, for example, the 1 million point set here, we actually see quite a bit more structure right here in the blue area, quite some more fine detailed structure, which is a bit counterintuitive, right? So we have less data, but we see more structure. So now the idea is, or the question is, like, can we do something with this? And if you need a t-SNE for, say, or a UMAP for a plot in a paper, then you need to create a single 2D static embedding, right? So there's not much you can do about this. But when you have this in an interactive exploration system, well, we can actually think about building a hierarchy on the data, right? So I would take my data and then in this illustration, build a hierarchy of 2 levels, right? So just for illustration, can also be more levels in the hierarchy. And then I can actually start exploring from this more abstract representation of the data, right? So I can just embed these points. And then if I want to have more details, say on the blue points here on the bottom right, I just make a new plot where I go a level lower, a level more detailed in the hierarchy, and embed these points again there. And of course, I could do this, right, for all of these points. Now, the question when we do this, when we build this hierarchy, is actually how do we build this hierarchy right? So, because if we just randomly sample points out of there, we run the risk of losing important small groups of cells, right? So rare cells that we're really interested in, they might be just removed by downsampling. So we wanted to make sure that this doesn't happen. And we also need to make sure that when we have this high-level abstraction of the data like this, that the structure that the t-SNE then embeds into the 2D space is actually still representative for the original data, and that we don't introduce like structure in there that is not in the original data. So I want to give a brief overview of how this building of the hierarchy works. So the idea is that we aggregate points by what we call landmarks that are just representative points that represent a small area or a small group of points around them. So we start here in this illustration with our original data, and we take these landmarks, right, in the lowest level, in the data level. This is just all of the data points. And we build a neighborhood graph on these. Now in t-SNE or other methods, right, we use the KNN. So we just use the same here to build the neighborhood graph on the lowest level of the data, right, on the original data level. But then when we go up in the hierarchy, right, so first of all, now we have to select our landmarks, right? So we don't want to randomly sample. So instead, what we do, and I'm going to spare you the math details, but in principle, we just select those points that are very well connected in the local neighborhood of these graphs, right? So we would look at this graph and then see, okay, there's a lot of connections over here, right? So then we basically want to take one of these points as a landmark. See? And now we have to build the new neighborhood graph, right? So we want to build the neighborhood graph now on this structure. But if we would just use a KNN, well, then we might connect these 2 points, right? But in the original data, they are not connected, right? So you really, if you want to go from here to here, right, you have to go around here. And this structure we really want to preserve now if we go more and more abstract. So we have to come up with a way to actually calculate a graph on this that represents this structure much more than just a neighborhood graph. So what we do is we basically now calculate what we called the area of influence, which is basically all of the points that are sort of in this vicinity or under the influence of these landmarks. So the way we calculate this is by a stochastic process, actually looking at like which points can be reached from each point, but I think the details are not that important here. But ultimately what we get is we get sort of what you see in this illustration here, sort of Hölts around each landmark, right? And then the similarity between two landmarks we can represent by the overlap of these, right? Because as you see here, maybe, right, so this group of points and these are overlapping groups of points. So when we have overlap in these areas of influence, then we say we connect the points in the graph, and then we get something like this. Right, so now we get a graph structure that is much more similar to the original graph structure, but we have many, many fewer data points in there. Right, so this really now retains this nonlinear structure in the data, which is really what we were looking for. Okay, yeah, so very quick demo of this again in Spatial. So basically here you see that same 5 million cell data set that I showed you before, but on this highest level of some data hierarchy. And basically now what I can do is I can just go in there, I select a group of cells. Actually, these are already clustered, so I just have to right-click in there and say zoom in. This is a group of innate lymphocytes. So now I say zoom in and I get a new representation of just this data, yet with more information from the hierarchy. So I'm going down and getting more detailed in the hierarchy. So I'm adding data points, but I'm only looking at the small area of interest there, right? And now maybe in a second, I'm going to do the selection like on top here. And then, yeah, so now you see actually that these three clusters here that you can see here, sorry, over here, they really represent to the three clusters you see correspond to these three clusters, right? Yeah, so that's a bit how you can then work with this directly in something like Cytosplorer. Now, the way I just presented this, one of the problems with this is that we basically always do this. Okay, I need to select something somewhere, and then I get a new plot that shows me more detail. Yeah, just like you just saw there. Well, we have implemented this in a way that it's basically very streamlined, but you still end up with a large amount of plots where you have your data in. And we published something on this in IEEE VUES in 2017 or 18, but then actually we took a step further and thought, why not actually try to keep stuff in place? So this is the standard exploration pipeline you see on top. But now in what we call the focus plus context exploration, we actually start again on this hierarchy, on this most abstract level of the hierarchy, but then instead of selecting, getting a new plot that only has the selection and so on, we would now say, okay, we do the selection over here, but then in place, we get the extra detail from the hierarchy. And by doing so, right, we're adding more points here that automatically pushes the rest sort of to the side in the focus area, while we get more detail in this area that we're really interested in. And we can do this multiple times, right? Again, I'm now doing selection here on the green part, doing the same again, and now I get, right, again, more detail over there. Right, and now what's really nice when we use this for imaging data is that, right, in this plot, I only have part of the image, but in this plot over here, I actually can see the whole image in one plot. Right, so this kind of focus plus context approach is really nicely fitting to these embeddings, because in the embeddings we only preserve local distances anyways. So it doesn't really matter if we sort of distort these axes, the global distances, right? And that has very little impact on the interpretation of the actual clusters that we see, right? And by modeling then this distortion directly in the embedding process, right, we get this very nice combination of different levels of detail, right, that we can add from the hierarchy. Okay, I'm going to show a very quick demo again because we're getting close to time, I guess. So again, this is a geospatial, or actually not geo, this is the sun. So we have a hyperspectral image of the sun here, And we are basically looking at mainly two clusters over here, right? So one is the background, one is the foreground, again, using sort of our recoloring trick. And now I can just select in here and then do sort of this process where I'm going to zoom into this piece of data in the hierarchy, right? So now I'm adding all this detail from the hierarchy to show actually much more detail in this group. And you see also how the detail now is added in the coloring, because this group just takes up more space in the color map, right? And doing the same again for a different group of sort of now the corona of the sun over here. Right? And we're slowly getting more detail, while also on the bottom here, that context area is being more and more compressed, giving less and less detail in this area here, actually. But of course, we can now just go and say, I actually want to have detail here. So now I will go to the lower cluster and set this as the focus, and you will see how everything else will just collapse. And this will take sort of the majority of the space in our embedding. Right, so now you already see the structure appearing here. You see the structure on the outer part slowly going away, right? So now all of a sudden we really see much more detail here on these areas, for example, on the surface, right? And again, one of the really nice things now is really that we always keep the whole image in this embedding. Okay, so I need like 2-3 more minutes. I just
Absolutely. If you want to take 5 minutes, that's perfectly fine. No
okay,
rush. Yeah.
great. So because I just want to show one more thing here now in really on this image space, and that is right now we did all of the exploration right really through the embeddings. But if you think about image exploration, right, you would actually have an image like this. You want to zoom, right? You would pan in the image. You might want to do annotations in there, but you mainly interact with the image and not so much with the embedding in this case, right? So again, with Alexander, we looked into actually how can we steer the exploration of these hierarchical embeddings, right, directly through the image interactions. So the idea is the embeddings we have, have rather limited resolution, right? What I mean with this, we do not want to cram too many points in there. Otherwise, we lose sort of these fine details. But this hierarchy level that sort of steers this is currently defined in the embedding. So the goal of this would be to drive this automatically, both the data that we show as well as the level of detail for the data through the exploration in the embedding, right? So we would use the available space in the embedding and the viewport of the image to find the right level of the hierarchy, and only that data that we're interested in. Right? So if you have an image like this, right, we built our hierarchy on this, right? So that would look something like this, very simple, right? So we have these 3 levels of the hierarchy here. This is our most abstract level. And then the embedding of this abstract level might look something like this, right? So we have maybe man-made structure and nature in there from this image of forests, roads, and buildings and so on. Now, if I want to zoom in, say in this area, right? So I would select the corresponding point in the hierarchy. I would also select the corresponding regions in the image, right? But now getting more detail would mean I move down in the hierarchy, get everything that's related to this, and then create a new embedding that shows this, right? Be it as its own image or in this focus plus context approach. But the general idea is always we go from top to bottom in the hierarchy. Now, if we look at images and steer this with the image, we actually have to reverse this. So if I do make a selection in the image, the way that we're doing is now we're looking, okay, which pixels are actually covered by this area, right? So this might be these pixels in our data hierarchy. And now to find the right level of detail to show this in the embedding, we just traverse the hierarchy up until we find like an amount of landmarks that would give us good resolution in the embedding space, right? So now we get an embedding that still has man-made structure and natural structures, but on a higher level of detail than just going here from the top. But it's also a selection of points that we couldn't get if we would start at the top, right? Because if I select this one and expand on this one, I always get both of these. So just getting one of these here on each side would be impossible in the old approach, right? So here, really, our direction is always going up. Okay, so then I can show this demonstration here, and that brings me then also to an end. So what you see on the left is the Washington Mall that I illustrated in the beginning of this section, right? And here's the embedding of this. And again, this is in hyperspectral satellite imaging data. So again, we use our coloring trick, right? And now if I do a selection over here, right? So then I see that is, for example, this area, but I can also go the other way around, right? So now I do a selection in the image space. You see it's basically distributed everywhere. So now I zoom into this one, and then our embedding automatically updates to match this selection. I can also pan, right? And you see some of the gray areas here appearing. These are points that were not in the embedding in the before panning, right? So here's a few more on the right side again. So we just source these, add these back to our embedding, right? And then recalculate the structure. All right, and I think this brings me to an end. So thank you very much again. And any questions that we still have time for?
Thank you so much, Thomas. What a cool presentation.